Hello everyone, it's wonderful to be able to connect with you. First of all, let me introduce myself. I am Dr. Magdline Henry Sum, and today I would like to take a little bit of your time to listen to my brief sharing that I have on a little project that we did here in University Malaysia Sarawak on the development of a competitive dot plot assay for detection of Reis-Tung-Grow disease. Well, you may ask me, what is Reis-Tung-Grow disease? Reis-Tung-Grow disease, or RTD for short, is unarguably one of the most destructive diseases of rice in South and Southeast Asia, including Malaysia. In severe cases, tung-grow susceptible varieties infected at an early growth stage could have as high as 100% yield loss. And the disease is caused not by one, but by two different viruses, namely Reis-Tung-Grow bacilliform virus, RTBV, and Reis-Tung-Grow spherical virus, RTSV. RTBV is a double-stranded DNA virus with a bacilliform structure, while RTSV is a single-stranded RNA virus with a spherical or icosahedral structure. And these are vector-borne viruses. They are transmitted by leaf hoppers, especially the Nephothetix malayanus and the Nephothetix virusans species of the green leaf hoppers. Plants that are infected by these viruses will show the following symptoms. First, it will have leaf discoloration, and this discoloration ranging from light yellow to orange yellow, and usually it will start from the tip of the leaves, and the leaves will be slightly rolled outward and twisted. And the plant will also be stunted in growth, and they also have reduced number of tillers and delay flowering. Delay flowering will lead into small panicles or panicles that are not completely exerted. And these panicles are usually sterile or partially filled grains. And all these symptoms will lead to low or no yield at all. One of the major problems with RTD is in diagnosing the disease or in detecting the disease in the field. Well, RTD is commonly detected by visual observation of the symptoms. However, visual identification based on the symptoms alone is not reliable and often confused with other diseases with very similar symptoms or even to non-pathogenic disorders, for example, soil toxicity or nutrient deficiency. Although currently there are different molecular techniques such as PCR, multiplex RT-PCR, or even real-time PCR used to detect the disease, these molecular detection are considered costly and require specific equipment and well-trained lab personnel. Another method of detection is by serological assay, and also have been shown to be specific, sensitive, and reliable. However, the limitation of these assays are on the availability of reliable reagents, and also, just like molecular detection, require laboratory facilities. Well, of course, one of the very obvious problems with this situation is none of these methods are suitable for field applications. These methods require specific equipment, well-trained personnel, which are often unavailable in rural areas where rice fields are situated. And this situation is especially true here in Sarawak, where most of the rice fields are located in rural and remote areas. And such lab facilities are only available at laboratories which is located far from the location of the fields. And this situation presents a few problems. And two of the major problems are, firstly, logistic challenges. This is to bring the samples from the fields to the lab facilities without compromising on the sample condition, because degradation can occur over a period of time. Secondly, the distance can cause delay in communicating the results, and that may lead to improper or no control measures. Therefore, in this project, our goal is to develop a simple, rapid, and reliable assay for detection of RTD that can overcome these limitations. And the ability to detect the disease on site will help quicken the decision making to carry out appropriate control measures. In this study, we developed an assay based on the dot blot assay platform. Dot blot assay is a cheaper, easier, and faster technique to detect the presence of protein in a biological sample. And it works based on the immunodetection principle for identifying specific protein. And generally, in the assay, protein is immobilized on the membrane, followed by successive incubations with primary and secondary antibodies that carry a tag to allow visualization. And in a competitive immunoassays, the number of antigen binding sites is limited by the competition of an inhibitor and a competitor. In our assay, our dot blot is designed to detect the presence of tungro in rice plants by exerting the antigen present in plant sample, which is the inhibitor, and the inhibitor competes with the recombinant antigens of the tungro viruses, which we will know as competitor, for the binding with corresponding antibodies. And any free antibody, meaning the one that has not bound to your protein of interest, will bind to the competing antigen, while the protein-antigen complexes will be washed away during the protocol. And the amount of antibody that binds in the well is an inverse measure of the amount of protein of interest in the original sample. The competitive dot blot assay that we have developed is an easy assay. The assay consists of 5 steps. Firstly, the antibody is incubated with the sample to allow primary binding of the antibodies to form antigen-antibody complexes. Then, the blot, where the protein have been immobilized, is added into the mixture. And what happened during the incubation is the unbound antibodies from the antigen-antibody mixture will bind to the recombinant protein on the blot. And after the incubation, the blots are subjected to washing procedure. Then, the secondary antibody conjugated with HRP enzyme will be added to the blot, and color is developed by addition of chromogenic substrate. Prior to the assay, the leaf sap is prepared from the plant leaves by grinding. And this whole process is designed and optimized as an on-site detection. And the result we expecting is the more antigen present in the sample, the less antibody is available to bind to the recombinant antigen on the blot. Therefore, if the mixture is prepared with plants infected with tungro, the blot will deposit no color. In contrast, color will be detected on the immunoblot with healthy plants. So, how is our assay differ from what is currently available? Well, let me tell you, one of the major advantages of this assay is the use of recombinant proteins as the antigen, not only for antibody production, but as well as to be used in the assay itself. These overcome the limitation in limited reagent supply faced by serological-based assay, especially for RTD detection. And another advantage of this assay is the reagents are stable in domestic refrigerator, which is about 8 to 10 degrees Celsius. Thus, it does not require ultra-freezer temperature for storage. And of course, the fact that it's easy and simple to perform. Who can beat that? This enables even the community to do it themselves. In conclusion, this assay that we develop here in University of Malaysia Sarawak does not require any specialized equipment nor a skilled personnel. And with this simple, rapid, cheap, and reliable assay, now everyone can do it. This is a great advantage for diagnosing RTD in rice field, especially for rural and remote areas. With this, I end my sharing. I thank you for lending me your ears. I bid you adios till next time.